Hi and welcome. I'd like to thank the organizers for inviting me to give this talk on genetic investigation in fetal CNS malformations, the old and the new, and I hope you'll enjoy the talk. So brain abnormalities are one of the most common congenital anomalies, and the incidence is uncertain because many are detected only postnatally, but the estimation is about 1 in 100, so about 1%. Of course, most of them are not that severe. So in the past, the genetic analysis of such brain abnormalities depended on karyotyping and then later on chromosomal microarray analysis. And these have long been the first diagnostic steps. So if we look at the prenatal diagnosis of all fetal malformation, and this is taken from a paper by Best et al., the estimation is that chromosomal microarray with or without karyotype may detect up to 35% of all fetal abnormalities, leaving about 65% undetected. And this is due to the fact that many are caused by single gene mutations, which cannot be detected by chromosomal microarray. If we look at the diagnostic yield of chromosomal microarray in prenatal diagnosis of brain abnormalities, we note that the detection rate of chromosomal microarray is quite low. It's only about 16%. This is taken from 2 studies. And the reason for that is probably there's a higher rate of brain abnormalities that are caused by single nucleotide variants, so mutation in hundreds of different genes. And this requires using advanced technologies like next-generation sequencing. So if we look at the studies showing the diagnostic yield of exome sequencing in prenatal diagnosis of brain abnormalities, we see that the rates hover about 22 to 34%. This study by Lord et al. Is quite an outlier because they had used a restricted panel of 1,600 genes and not the entire exome, and this may be one of the reasons. So we have been collecting for a long period of time samples from fetuses that have undergone termination of pregnancy due to severe brain abnormalities. And then when next generation sequencing became widely available, we started employing that first on a retrospective basis. So we started off with 114 fetuses that had such severe brain abnormalities that warranted termination of pregnancy, TOP. Cases with mild ventriculomegaly were usually not included because they had not merited such termination. All cases were first analyzed by chromosomal microarray, and the ones that were chromosomal microarray negative also went whole exome sequencing trio analysis, and this was published in Ultrasound Obstetric Gynecology a couple of years ago. So first, let's look at the results of the microarray analysis. So starting off with 114, we found 11, so 10% of abnormal chromosomal microarray results. And these were all over the place, and it resulted in different brain abnormalities. Most of them had been isolated, but some had other manifestation like intrauterine growth restriction and short lobe bones, and this one had hyperechogenic kidneys. So when we look at the on the chromosomal microarray results, we see that most cases were deletion, except this case here was deletion duplication resulting from a unbalanced translocation, and this one case here was a triploid. Then for the cases that were chromosomal microarray negative, we went on to doing exome sequencing. And our results show that of these cases, we had 86 cases that underwent exome sequencing, and of these, we found positive results pathogenic or likely pathogenic in 38, so that's 44%, which is higher than what has previously been reported. We were quite happy about that initially. And the results were not usually were as expected. For instance, for aqueductal stenosis, we detected an L1 mutation, and cases that resulted in hemorrhage had collagen 4A1, collagen 4A2 or A1 mutations. And then DCC may explain corpus callosum abnormalities and so on. But some cases were a little bit surprising. Of course, this series here of malformations of cortical development had the expected suspects like tubulins and so on, but also a collagen 4A1, and this has been also shown in other studies as well. So this has led us to the thought that regardless of what type of brain abnormalities, we wouldn't focus on a panel of mutation as wide as it could be, but rather do a whole exome sequencing and look at all the genes that are relevant. So when we divided the cases according to the clinical category that was defined by the ultrasound team, the detection rate for chromosomal microarray for multisystem was 13, but for whole exome sequencing was 44. For complex brain abnormality, was 16 for chromosomal microarray and 58 by whole exome sequencing. And then we had very little detection for chromosomal microarray for other categories, but they were quite high for most categories using whole exome sequencing. When we looked at the mode of inheritance of these abnormalities, we found that the majority of them were actually inherited, either X-linked, recessive, or dominant from an apparently unaffected or mildly affected parent, and slightly less than 50% had been de novo. So this has significant implications for patients, and indeed some of the patients with recessive or X-linked condition went on to undergo pre-implantation genetic testing at our center to eliminate the recurrence. So to sum up, when we looked at our first series of severe brain abnormalities undergoing termination of pregnancy, we found that copy number variants accounted for 10%, monogenic for 44%, leaving about 46% unknown. Then we compared the cases that had been recurrent. So we had some patients who had the same abnormality recurring. And we looked at the diagnostic yield of whole exome sequencing in recurrent cases versus first-time cases. And we saw a significant, although not unexpected, higher yield in those cases that are recurrent, because this would obviously mean that we're dealing with a genetic abnormality. Then we asked ourselves whether chromosome microarray is actually required in the workup of CNS abnormalities. Because the diagnostic bioinformatics pipeline that we use is called Franklin by a company called Genox, detects copy number variants ranging from even single exons, of course, to entire chromosomes. So what we did was we reanalyzed all the cases that were positive, that had positive copy number variants, and then we reanalyzed them by whole exome sequencing, and we were able to detect all 10 pathogenic copy number variants using the whole exome sequencing. And we had additional copy number variants that were benign, but still were detected in these cases. And these two were detected by whole exome sequencing. And the size of these copy number variants range from as low as 335 kilobases to 19,000 kilobases. So at least for the workup of termination of pregnancy, we think that whole exome sequencing is sufficient. And then when we have a chance and we have the funding to do the genetic workup of brain abnormalities, we go straight ahead to whole exome sequencing, because we assume that we can detect all single nucleotide variants, all point mutations, et cetera, but also copy number variants. So this sums up our results. We for the entire retrospective series, beginning with 114 cases. 11 were detected by chromosomal microarray, and then except for the one that had insufficient fetal DNA, this was also confirmed later by whole exome sequencing. And then the ones that were negative, we offered exome sequencing, some declined, some had insufficient DNA, but then when whole exome sequencing was performed for 86 cases, we had 44% detection rate. So that was a total of 96 cases that were analyzed retrospectively by whole exome sequencing. So to sum up these results, if we had only done exome sequencing, we would have detected 54% of the cases using exome sequencing, leaving still 46 cases unknown. Then we had decided to go ahead and do a prospective study, and now including both fetuses that had severe abnormalities undergoing termination pregnancy, but also cases that were, at least at the time, were ongoing. So these included 89 fetuses, 38 with severe abnormalities undergoing termination that had undergone whole exome sequencing, and 51 undergoing ongoing pregnancies. With ongoing pregnancy, we still do chromosomal microarray because this is the demand of the Ministry of Health. And of course, the CMA negative cases underwent whole exome sequencing. So without going into too much detail, because this maybe a little complicated, we show this here. So in retrospective termination of pregnancy series, we had a yield of about 50%. In the prospective series, the case that had undergone termination of pregnancy, we had a diagnostic yield of only 24%, and for the ongoing pregnancy, only 14%. And this had us a little bit confused until we understand that we had lowered the bar. So in the retrospective series, as I've shown you before, we had many recurring cases. And of course, this increased the diagnostic yield because these are more likely to be genetic versus the prospective study that we hadn't had any of these recurring cases. And in the ongoing pregnancy, these were ongoing because they were not as severe and included even mild brain and anomalies that they may actually be normal variants. And this may explain why our yield has appeared to be lower. Nonetheless, we had interesting results. So for the multi-system abnormalities in the prospective studies, we had a detection rate of 33%. And of course, we had detected both mutations. We'll talk about this NDUFF5 case a little later. And then we also found some microdeletions. And of course, we had other abnormalities. This was an interesting case that appeared to be an encephalocele caused by a TMEM231 mutation. So I'd like to talk a little bit about the importance that we find in the workup of variants of unclear significance. So when you do whole exome sequencing, you find often more than 100,000 different variants. So you have to analyze them according to specific guidelines, usually by the American College of Medical Genetic Guidelines. And still, some variants appear to be significant, but they don't pass the bar, don't pass the criteria for being called pathogenic, and therefore we're not sure that they are causative of the abnormality. So I'd like to describe to you a case that we found the workup interesting and important. So this is a case of an Ashkenazi Jewish couple. They had a previous pregnancy terminated because of posterior encephalocele and large kidneys. And then in the index pregnancy, they had an early anatomy scan as early as 14 weeks that showed extremely large kidneys with cystic dysplasia in abnormal cerebellar morphology. And as you can see here in figure B, that the brainstem has an abnormal morphology, and that and also here in C, you can see a very dilated fourth ventricle communicating with the cisterna magna. So obviously this was a severe case. When we did exome sequencing, we found that the fetus was compound heterozygous, meaning that they had two different variants in this gene. One was a clearly pathogenic variant inherited from a normal mother. Of course, it's a recessive condition. And this change results in a stop codon, so it's obviously pathogenic. But the paternal variant was intronic. It was in the intron, and it was a deletion, but it was quite distant from the splice site. So it was between -12 and -16. So of course, this was suspicious because it fitted the phenotype, but of course, we could not determine at the time that this variant was indeed pathogenic. So what we did, we ran RNA analysis. We took the father's cell, and then we extracted RNA and did RNA sequencing. And the RNA sequencing actually told the tale, because what happens is that this deletion, creates a cryptic splice site in the intron. And therefore, what happens is that this cryptic splice site adds additional 22 base pairs to exon 13. And this has been shown in sequencing as well. And what happens is this causes a frame shift and a premature stop codon. So we were able to then determine that this intronic variant that was initially considered as a variant of unclear significance is actually a likely pathogenic variant. And then we had cases that we were able to use RNA sequencing to actually rule out the pathogenicity of certain variants that appeared to be initially as causative pathogenic. This other case shows the importance of sharing data information. So this involves the gene that I referred to before, NDUFF5. And this is a result of shared information between 3 medical centers in Israel. This was made possible by using the community mode in the Franklin pipeline, which allows you to see whether other peoples have seen similar variants in the same gene. Okay, so the first case was from the Rabin Medical Center. The first fetus they had was at 11 weeks, had bilateral cysts disrupting a normal mid-brain architecture and compressing the lateral ventricles, and also had pleural and pericardial effusion and other abnormalities, and subsequently hydrops fetalis with nuclear edema and enlarged heart, and they underwent termination of pregnancy. And then the second fetus that they had similar malformation with brain cysts and hydrops fetalis and pleural and pericardial effusion, underwent termination of pregnancy. Then when they ran the exome sequencing for the second fetus, because the first one was unavailable, they showed a compound heterozygous variance in this gene. And of course, just like I showed you before, the paternal variant is likely pathogenic because it has been seen before in affected individuals, but the maternal variant was a variant of unclear significance. It was a missense. So they weren't quite sure about how to see whether this was causing the abnormality. Then when they did some of the work, they have been able to show that there was a decreased amount of mitochondrial, and the mitochondria appeared to be abnormal. So this was in line with NDUFF abnormalities. So it seems to be associated with this gene. And then they were able to see that at about the same time, we had a second family that had 2 abnormal fetuses. So the first one was detected at 17 with agenesis of the corpus callosum and also cystic cavitation of the ganglionic eminence with abnormal lamination, fetal growth restriction, again undergoing termination pregnancy. And the second pregnancy that had was actually a triplet pregnancy, but one fetus already demonstrated the same abnormalities and undergone multifetal pregnancy reduction. And using the whole exome sequencing analysis, we show that both were homozygous for the same NDUFF variant. And both parents, of course, were heterozygous carriers. And because of this variant, was actually the same variant that was detected as a compound variant in the other case. So this appeared to be a causative in our case as well. And about the same time, there was another family from another hospital showing, again, similar brain abnormalities, but that was detected earlier. They had shown different abnormalities, but again, they had the same paternal mutation in the first case. So this shows you that sometimes when you see variants that appear to be causative, it's important to share your information and make benefit of other people's information that we're sharing. The other case I described in the first talk that I had given about malformational cortical development, this just showed a case that had significant macrocephaly that had been demonstrated at a very late in pregnancy, because at 28 weeks, the head was large, but still within normal range, but at 35, it was plus 3.4 standard deviation. Of course, pregnancy was terminated, and the MRI showed significant abnormalities with the macrocephaly, abnormal operculation, and abnormal sulcation pattern with frontal polymicrogyria. And it was suspected that this was caused by the mTOR pathway, but then when we ran whole exome sequencing on the fetal samples, we actually overlooked this variant here because it was very low rate. But then when we took brain samples and we ran the brain samples, we were able to show a higher incidence of this variant, so about close to 20%, and you can show this variant here. So, of course, it's important to recognize that the fact that we detect only about 40 to 50% of cases with such severe brain abnormalities, this is because we do the analysis usually on fetal blood or amniocentesis samples. But it's important to keep in mind that a lot of cases may be caused by somatic mutation, and these are post-zygotic somatic mutation. For instance, in our case, we found this PIK3, the mTOR pathway mutation. And these may cause both hemimegalencephaly, but also macrocephaly and other abnormalities. And this requires taking a sample. It is challenging, and I can't figure out a way of taking a sample in ongoing pregnancy. But of course, when we analyze these cases postnatally, then we can, it's important to take note of this possibility. And it's actually important for the parents to know, because if it's a post-zygotic mutation, the recurrence risk is actually very, very low. And so with this, I complete. I'd like to thank all the collaborators that help with this analysis, the team at the Genetic Institute, headed by Professor Boris Feldman, the molecular laboratory headed by Anat Bashira, and the Genome Center headed by Adi Morin. Of course, our ultrasound unit, previously headed by Gustavo Malinger, now by Professor Nina Kreiden-Aratz, and the entire team, and also the imaging department with Liat Ben-Sira on MRI. Thank you very much.